Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Optimizing Monoclonal Antibody Production, Temperature and Osmolality Impact. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Advanced Instruments who developed the content for this presentation. Advanced Instruments is a global company offering a novel portfolio of analytical tools, including Osmotech, a robust line of microosmometers to support bioprocessing and quality control, and Celentum, a portfolio of best-in-class imaging and single-cell deposition technologies for cell line development workflows and assurance of clonality for regulatory bodies. Now I'd like to introduce our speakers for today's event. Rick Zampa has been with Advanced Instruments for 27 years. During his tenure, he has assumed roles in sales, customer service, and technical service, and has been actively involved in application support, product development, and marketing. His current role involves application support for their portfolio of biotech osmometers. And Shazana Muhammad started her PhD research in late 2021 at Imperial College London under the supervision of Professor Cleo Kontaravdi, investigating the generation of host cell proteins in the upstream and their subsequent clearance in the downstream processes. But now, without further ado, let's go ahead and hand things over to our first speaker for today. Rick Zampa, you may begin when you're ready.
Thank you for the introduction, Ryan. Before I hand over to Shazana, I want to give some background information on Advanced Instruments for those who may not be familiar with our organization. Advanced Instruments is the global leader in osmolality testing and has been for over 65 years. Along with this portfolio, we are also experts in cell line development technology and liquid handling quality assurance. Our products are dedicated to increasing productivity and assuring quality drug product in the biopharma sector. Our headquarters are in Norwood, Massachusetts, with offices in Westbrook, Maine, and Winbourne, UK. We have an ever-growing install base in over 90 countries globally, with the latest direct sales teams in France, Germany, Austria, and the Benelux regions. These are complemented by our worldwide distribution network. Advanced Instruments currently offers 4 different product lines that are utilized in the biopharmaceutical space. Today, we will focus on the Osmotech osmometers and the various applications in upstream processing. Osmotech is the only osmometer portfolio that is specifically designed for customers in the biopharma market. The instruments are optimized for testing of biological samples such as cell culture media, buffers, and aqueous drug products. We also offer the Celentum instruments and reagents for cell line development workflows, with devices focusing on single-cell seeding and whole well imaging. We also have the Inoximet anaerobic jar system for bacterial cultivation and the Artel instruments for pipette and automated liquid handling calibration. All 4 product portfolios are supported by our own dedicated Advanced Instruments trained field service engineers. And we have service contract options available to help you maintain the equipment while preventing any disruptions to your workflow. Specifically for our Osmotech users in biopharma, we offer qualification and validation services to ensure regulatory compliance while allowing you to bring your instrument online quicker. Here, I want to focus more on the devices that can be positioned into the upstream segment of bioprocessing workflow that we'll be talking about today. The Osmotech osmometers are built with biopharmaceutical labs' needs in mind, and were designed as the result of our customers' feedback. For our Osmotech Pro and our Osmotech HT, we provide a range of up to 2,000 milliosmoles. With our Osmotech XT device, providing osmolality testing up to 4,000 milliosmoles, which is perfect for those higher concentration or more viscous solutions. The testing process is fast, and each is simple to integrate into any customer workflow and throughput of testing. They have flexible data connectivity options and strong data integrity features, which are adjustable to your lab's specific needs and are therefore flexible for R&D, process development, and manufacturing settings. The Osmotech HT, launched last year, is the first 96-well plate-based osmometer on the market and is suitable to those high-throughput automated workflows we commonly see in not only upstream QC, but also within formulation development. Also, the Osmotech portfolio of microosmometers currently support the different regional pharmacopoeias such as European, US, Japanese, and more. If you would like to learn more about our osmometers or any of our other portfolios, my contact details will be provided at the end of the presentation. With that, I shall pass over to our guest speaker, Shazana. Thank you.
Thank you, Rick. Hello, everyone. Thank you for taking your time for this webinar. In this webinar, I will discuss the optimization study that I'm currently working on that involves monoclonal antibody production, specifically focusing on the impact of simultaneous change of temperature and osmolality in a cell culture. Monoclonal antibodies, the predominant class of new drugs, high approval rate compared to the other drugs. The market value is expected to reach $300 billion next year. I believe many here are already familiar with monoclonal antibodies, but just a little bit of introduction of these proteins. They are generally monospecific antibodies that are extracellularly secreted by mammalian cells. These antibodies have various applications, especially as therapeutic drugs for viral disease, cancer, and recently, they can possibly be used for antivenom. How do monoclonal antibodies function? In the case of pathogens, they bind to the antigens on the surface of the pathogens and neutralize their ability to infect human cells, or they tag them for elimination. Now, to produce these antibodies, we need to use some producer cell lines. One of them, which are widely used for manufacturing, known as Chinese hamster ovary cells, or CHO cells. In the past 15 to 20 years, scientists and engineers worked hard to increase the antibody yield, and through the improvement of process technology and cell engineering, the antibody yield could now go up to 10 grams per liter. However, as the yield increased, the number of impurities also increased. Among these impurities are the host cell proteins. These are the native proteins relieved from the CHO cells throughout the cell culture duration, majority at the late stage due to cell lysis upon cell death. They also can be secreted with the product. These host cell proteins were mostly removed during protein A purification, and the level can be reduced to standard requirement of 100 nanogram per milligram of protein. But there are host cell proteins that possibly remain in the product at the end of downstream process, which are then categorized as difficult to remove host cell proteins. Some of the impact that could rise from these remaining host cell proteins are they can pose immunogenic risk to the patient. They can even be fatal. For example, phospholipase B-like 2 or PLBL2 protein. This protein was found in lebrikizumab clinical material. From the clinical studies data, this PLBL2 was reported to cause specific and measurable immune response in around 90% of the subjects. While continuously passed throughout the downstream process with the product, these remaining host cell proteins could alter the antibody structure. Some of host cell proteases can naturally break down the peptide structure bond of the antibody, and the host cell protein bindings that play important role in proper protein binding in endoplasmic reticulum and Golgi could interact with antibodies and promote intracellular aggregation. This diagram shows a typical upstream process of monoclonal antibody production, where there are a series of bioreactors, the seed bioreactor for seeding, and production bioreactor for the main cell culture. The seed bioreactors are run through batch modes, where the media and cells are fed together in the bioreactor. While for production bioreactors, they are run either using fed batch mode, where nutrients are fed at certain interval throughout the cell culture duration, or perfusion mode, or known as continuous culture, where media is added continuously while removing the spike medium that contains the product and cells. Because these two modes of operation can increase culture longevity, which then increase the yield per batch. The typical duration of a fed batch is between 14 to 15 days, and perfusion culture can go up to 17 days. There are several parameters involved in the cell culture process. Some of them are identified as critical process parameters. These parameters need to be monitored and maintained within their specification because they can directly impact the critical quality attributes of monoclonal antibodies. For example, the cell culture media, the nutrient feeding, temperature, osmolality, dissolved oxygen, and pH. Therefore, it is very crucial to optimize this parameter to ensure a robust and reproducible process. As we know, manufacturing processes are run for a number of times to fulfill the market demand. The process optimization is also important when we want to scale up to manufacturing scale or when we want to scale down to perform process validation. One of the critical parameters in the cell culture is the temperature. Temperature can directly influence the cell growth, cell viability, and cell metabolism. It can also affect the synthesis of monoclonal antibodies. Generally, the temperature is set at 37 degrees, which is the physiological temperature of cells. And now, the typical working temperature can be between 31 to 37 degrees. The low working temperature, or also known as temperature downshift strategy, the TDS, it is performed where the operating temperature is reduced to hypothermic condition after certain days of cultivation. The purpose of this strategy is to control cell proliferation and lowering glucose consumption and oxygen uptake rate, thus reducing lactate and ammonia production. Through this strategy, the cell culture duration can be extended and the antibody production can be increased. In this low working temperature, the sensitivity of cells to apoptotic agents is minimized, allowing the culture to be harvested at high cell viability. Recent report found that the hypothermic condition could influence the whole cell protein content in the cell culture, where the cell cultures operate under mild hypothermia were found to have reduced host cell proteins concentrations. Another parameter, the osmolality, indicates the osmotic pressure condition of a cell culture. Since it controls the transport of water and nutrient through cell membrane, the level of osmolality can significantly impact the cell growth and cell metabolism. Essentially, the osmolality of a cell culture media should resemble the osmolality of the inner cell for the cells to be able to grow in that media. Depending on the culture medium, the osmolality level of a production culture can range between 320 to 440 milliosmol per kg. This level, however, can inevitably increase when extracellular metabolites accumulate during that stage culture. In a fed batch culture, the osmolality level can increase as a result of concentrated feeding media, and routine nutrifeeding can also lead to overfed culture. The common impacts of the high level of osmolality, or also referred to as hyperosmolar condition, are decrease of cell viability and cell growth rate. A recent finding demonstrated that a deterioration of glycan distribution profile in the cell culture that operates at high osmolality level. In contrast, some studies have reported several advantages of hyperosmotic condition. Other than it can enhance the nutrient transport into the cells, this condition could also indirectly induce the G1 phase arrest, where this phase was reported to be the major production of antibody, thus increase the antibody yield. Although there are contradictory findings to this, hyperosmolality at certain level in the cell culture could result to better antibody quality and biopotency. In addition to that, there are studies reported where simultaneous change of osmolality and pH could maintain the cell viability and resulted to lower protein aggregates. These findings show how the osmolality level could influence the cell culture performance, but could it also influence the host cell protein content in the cell culture? And to what extent the impact of changing these parameters on generation of host cell protein in overall? In order to answer these questions and to study which upstream condition involving these two parameters that could potentially minimize the level of host cell protein at the end of the cell culture, my study focused on investigating the impact of simultaneous change of temperature and osmolality not only to the cell culture performance, that includes integral viable cell density, IVCD, cell growth rate, and cell productivity, but also on the extracellular accumulation of the host cell proteins and how these host cell proteins were removed in the downstream process. This is the experimental design for my work, which was developed using Mode 12 software, where the range of the temperature was between 32 degrees, which is the hypothermic condition, up to 36.5 degrees, which is the physiological temperature of the cells, with a midpoint temperature at 34 degrees. While the osmolality level was set at 410 milliosmol per kg for the low level to a high level of 500 milliosmol per kg, with a midpoint at 455 milliosmol per kg. These parameters will change on day 5 of culture when the cells are at the late exponential phase, and these cell cultures will perform in duplicates for every condition. The control culture in this experiment is the culture run at 36.5 degrees with osmolality 410 milliosmol per kg, which is actually the osmolality level on culture day 5. This symbol shows the low, mid, and high level of each parameter, which will be used in some of the results later. Well, the experimental process where the cell culture run were in a small 25 ml shaped flask in a fed batch mode, where I perform feeding every other day until the cell viability drop below 80%. Then I harvest the cell cultures. I use 5 molar sodium chloride to increase the osmolality of the cell culture, and I monitor the osmolality level every other day using Osmotec XT. I measure the cell density and the antibody titer daily, and the host cell protein concentration was measured only on certain days. After harvesting the cell culture through centrifugation and filtration, I purified the supernatant using protein A column and further purified the product using cation exchanger. The antibody titer and host cell protein concentration were measured for both purification steps. The graph shows the integral viable cell density of all cell cultures. Integral variable cell intensity, or IVCD, is a measurement of active cells on day-to-day basis, where we can determine whether the cells are more active or less active at different conditions. The duration of all cell cultures range from 11 to 14 days. From this graph, it shows that the hypothermic cultures, or cultures that run at 32 degrees, denoted by R7, R8, and R9, they have the longest duration, which is here. At 36.5 degrees, which is plot R1, R2, and R3, the IVCD is high for all cultures, except cultures that run at midpoint osmolality R2, where the plot is here. Interestingly, at 32 degrees, which is the lowest temperature, the cells are more active in the cultures that run at 410 and 455 milliosmol per kg, which is the low level of the osmolality and the midpoint level of osmolality, as the IVCD of these cultures are among the highest, which is here. So it is almost similar to the control culture, which is here. The lowest IVCD is observed for cultures that run at the highest osmolality at 32, which is here, R9, and 34 degrees, which is here, R6. So the plot is here. They have the lowest IVCD. This demonstrates that the cells become less active when we increase the osmolality to the highest level. This graph shows the specific growth rate of the cells for different culture conditions. The average of the growth rate, which was calculated from day 5, where level of temperature and osmolality were changed, is between 0.11 to 0.14 per hour. The average growth rate for culture run at 36.5 degrees is on the higher range, between 0.13 to 0.14 per hour, while the growth rate for cultures that run at 32 degrees is on the lower range, between 0.11 to 0.12 per hour. At 34 degrees, the cell growth rate has similar pattern to the culture run at 36.5, only at midpoint osmolality level, where the cell growth rate is on the lower range. Nevertheless, the difference could be considered low, which could indicate that there is a positive synergy between temperature and osmolality. This is the graph for cell-specific productivity. From this graph, we could see that as the temperature decreases, the productivity also decreases. If we compare based on osmolality level, we can see that culture run at midpoint osmolality, which are these blue lines, the R8, R5, and R2 are here, have higher specific productivity. Meanwhile, the productivity for cultures run at highest osmolality level, which is R9, R6, and R3, is almost constant for all temperature. The plot can be seen from here, which is about in the middle of all lines. In overall, the range of the specific productivity for all cell cultures are actually quite close to one another, which is between 0.02 to 0.03 picogram per cells per day. For the whole cell protein concentration, I decided to measure when the cell culture viability started to drop to 95%, which typically on day 7. Then, depending on the duration of the cell culture, I measured every other day, 2 days before harvest, and on the day of harvest. I would like to recall back the temperature setting for this culture ID, where R1 to R3 operate at 36.5 degree, R4 to R6 operates at 34 degree, and R7 to R9 operates at 32 degree. From this chart, the concentration of whole cell protein increased from day 7 until harvest day for most of the cultures, which is possibly due to cell death. Interestingly, the hypothermic cultures run at 455 milliosmol per kg, denoted by R8, show a declining HCP concentration starting from day 9 of the culture. The other cultures with decreased whole cell protein concentration on the harvest day are the R3 and R4. These are actually the cultures that run at lower temperature, 34 degree, without change of osmolality, and the cultures that run at 36.5 degree with the highest osmolality level at 500 milliosmol per kg. The cultures run at 36.5 milliosmol. The cultures run at 36.5 at 455 milliosmol per kg, which denoted by R2, shows a decreased whole cell protein level on the harvest day. But this low level could correlate to the low IBCD results earlier on. Although with highest IBCD, the culture run at 32 degree at 410 milliosmol per kg also show a decrease in whole cell protein concentration on harvest day. This chart shows ratio of whole cell protein to monoclonal antibody for all cultures. In most of the cultures, this ratio drops as the cell culture progresses from day 7 until harvest day. At midpoint temperature 34 degree, especially the culture run at 455 milliosmol per kg, this ratio almost constant from day 7 until harvest day. The culture that run at 32 degree at 410 milliosmol per kg exhibit a steady decrease ratio of whole cell protein to mAb from day 7 to harvest day. And the ratio of this culture is also among the lowest ratio of whole cell protein to mAb on the harvest day. The lowest ratio of whole cell protein to mAb on harvest day is observed for cultures that run at 455 milliosmol per kg for all temperature, indicating a potential culture condition that could result with lower whole cell protein accumulation while maintaining good yield of product. And the R9 cultures seem a bit odd with a bit high ratio on day 9, then this ratio decrease and increase again on harvest day. The ratio is high on day 9 was because the antibody titer was quite low on this day, and it started to increase the day after and became constant close to the harvest day. This is the statistical results that show there is an interaction effect between temperature and osmolality on the ratio of whole cell protein to mAb on day 7 and on the harvest day. This is the results for whole cell protein concentration at different downstream steps, where obvious reduction of whole cell protein is observed for the control culture that run at 36.5 degree and 400 milliosmol per kg. The other culture show different of whole cell protein level is not apparent even after further purified through cation exchanger. Meanwhile, at this midpoint setting of temperature and osmolality, it seems that cation exchanger could not further reduce the level of host cell protein. From these findings, some of the key points that I would like to highlight are: there is an insignificant difference in average cell growth and production rate for all culture conditions. This could indicate positive synergistic effects of both parameters. Some culture conditions have the potential to produce a culture with lower host cell protein level and at the same time maintain the antibody yield, which in turn can lessen the load on the downstream process. Moving forward, I would like to further investigate the host cell protein removal in the downstream process at larger scale or the bioreactor scale, and then identify the species of the remaining host cell protein in the product. Thank you all. Now I will hand over to Ryan for Q&A.
Well, thank you so much for that very insightful presentation. At this point, I'd like to invite our audience to continue to send their questions or comments right now using the questions window for this Q&A portion of our webinar. Now, I've already received some questions for you, so we'll get ourselves started with those. Looking here, the very first question I have would like to know, are there use cases for osmolality anywhere else other than the upstream cell culture?
Hello, Ryan. Yes, thank you for the question. Yes, there are many different use cases for osmolality other than upstream. We have downstream buffer prep is one of the other applications, as well as formulation development and final QC, release QC. There's a number of developing applications as well throughout the bioprocessing workflow. And if you'd like to get some more information on that, we have a number of papers and webinars on our website you can take a look at. And if you have further questions, you can contact us about those.
Perfect. Thank you so much for that answer, Rick. The next question I have here, and I think it's for Shazana. I think what they'd like to know is, why did you use sodium chloride to change the osmolality?
Actually, I use sodium chloride because I want to minimize I want to minimize the variation of the results, and I want to exclude the factor that could impact the antibody itself, caused by earlier on when I explained on the overfeeding issue. So that's why I use sodium chloride, because I just want to see the increase of osmolality and how that impact the cell culture performance.
All right, very good. Thank you for that. Someone else is wondering, would the seeding density change the result of this study?
Okay, for the seeding density, actually that depends, but I believe it will not cause a major impact to the performance of the cell culture. But I would suggest that we do initial or preliminary experiment to see the growth first and to see whether it's linearly it's in linear profile comparing to my study. If it's linear, we can expect the growth will be almost the same. Because if you see that I made the changes or the manipulation on day 5 because I want to see first how the growth of the cells, and then I made the manipulation.
Very insightful. Thank you for sharing that. Now, Rick, I have a question for you, I think here. Someone's wondering if our solution is sitting at 300 m/0 what instrument would you recommend?
Well, it somewhat depends on the regulatory needs that you're in at this time, but we would usually recommend the Osmotec XT instrument for its full data integrity and compliance abilities. Alongside that, it has the best developed intelligent freezing technology, which we have introduced with the XT, which will give you the opportunity if your solutions or your drugs change in complexity or viscosity to allow you to test more complex solutions down the road. So it gives you some abilities to change your processes and move to different processes throughout the structure of your program.
Okay, thank you so much. Now, someone else is curious then, can you triplicate testing when using the automated Osmotec HT device?
Oh, yes, yes, you can do triplicate testing out of one well. The Osmotec HT is a 96 well plate format. So if you're looking at using the high throughput type of systems where you're doing plate-based testing, you can take that plate-based testing and move it into your osmometer. And you can get 3 tests out of each well for the Osmotec HT. And this will also help with the new changes to EU pharmacopoeia that have come into effect this year in January, which requires you actually to run triplicate testing on your controls and standards in the process now.
Perfect. Thank you so much. Now, moving on to another question here, Shazana, someone's wondering, do you use an ELISA CHO HCP kit, or did you develop your own method to capture the HCP concentration?
I used the ELISA kit from the commercial one.
Perfect, thank you. Next question here is wondering, what is the volume that you're looking at to scale up the experiment?
For the scale-up purpose, I'm looking at least 1 liter of 1 liter volume to run it, because I want to, because it will be processed to different purification. So I need enough sample to further evaluate and investigate the host hypothesis species in there.
Right, okay. So what would you say is your expectation for the scale-up outcome? And further, are the results going to be the same or different?
Actually, there are also other studies that did scale up from shake flask to bioreactor, and actually the whole cell protein content is quite similar from shake flask to the bioreactor, except for the species. They are different, and the different are quite impactful. I mean, it's very different from shake flask to bioreactor, so it's very important to see that also in my study.
That's wonderful. Great. Thank you so much for that answer and for all of the answers. However, I think we've reached the end of our time here today with our speakers. Now, if we couldn't get to your questions, know that the speakers will reach out to you after the webinar. Advanced Instruments will follow up with you. Or if you have some further questions, you should direct them to the email address that I have up on the screen. I want to thank all of the audience members for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link in the chat box to view the recording of today's event, which you can also share with your colleagues so that they may register for the recording here as well. Now, please join me once more in thanking our speakers for their wonderful time here today. We hope that you all found the webinar informative. Have a great day, everyone, and thank you for coming.
Thank you, everybody. Take care.